Hello, this starts Module 3. Module 3 is the beginning of our discussion of the central dogma. In this session, we'll talk about nucleotides as well as the synthesis of purine and pyrimidine bases. This is synergistic with chapters 12 and 41 in your textbook. The general aspects of this will be the following. First, we'll look at the structure of genomic DNA. We'll review some basic nomenclature as well as the superstructure of DNA as it exists in a chromosomal structure. We'll look specifically at the nucleic acid bases. There are 5 primary nucleic acids, the purines, adenine and guanine, and the pyrimidines, thymine, uracil, and cytosine. In general, nucleotides consist of a base, a sugar, and a phosphate group. Finally, we'll discuss the synthesis and the salvage of purines and pyrimidines. The following depicts the central dogma, where DNA can be replicated during the cell cycle. DNA, which encodes our genetic information, can be transcribed into RNA and translated into protein. The conversion of DNA down to protein is what is termed the central dogma. Over the next few modules, we will go through these processes, starting with DNA, its superstructure, followed by DNA replication. Next, we'll look at transcription and translation. This will be followed by specific regulation of these processes, and then a summary module looking at inheritance patterns of chromosomal DNA. The human genome consists of 22 homologous autosomal chromosomes and a pair of sex chromosomes. Here you see the chromosomal arrangement for a male. We can tell this because of the presence of both an X and a Y chromosome. Our genome has 2 major functions. First of all, it stores our genetic information. It is our master copy, and it is integral that we maintain an undamaged copy for genetic stability. Second, it's responsible for the direction of the transcription of complementary molecules. The initial complementary molecules will be RNA, which are translated into proteins. The chromosomes, or homologous chromosomal pairs, are arranged the following. They're arranged in genes, and genes are a linear stretch of DNA which encodes for a protein. If we look at a homologous pair, this would be one chromosome, and this would be its homologous chromosome. You can see that genes are aligned with one another. This would be gene A on one homologous chromosome, and this would be gene A on the adjacent chromosome. Each of these genes, although similar in nature, can be dominant or recessive to one another. Gene A, for example, this allele encodes for protein A, and gene A on this homologous chromosome also codes for protein A. These would be considered alleles that are the same. If we look at gene B, for example, although it's similar on these homologous chromosomes, you can see that it's slightly different. This lowercase b would be an example of a recessive allele. Again, it's the same gene, and it encodes for similar proteins, but this allele would encode for protein B, whereas this allele on the homologous chromosome would encode for a recessive gene, denoted here as lowercase b. This will be important in later modules when we look at the heritability of different alleles and how that can contribute to a disease process. Remember, a dominant allele is usually always denoted by a capital letter, whereas a recessive allele is denoted by a lowercase letter. Each gene is comprised of at least 2 alleles. If we look at a single chromosome, you can see, firstly, that it has a heterogeneous structure. All chromosomes have telomeres at the end, and these are repetitive sequences that are necessary during DNA replication and allow for the chromosome to be replicated up until the end without having shortening of the chromosomal end. We'll look at this in the next session when we review DNA replication. Then you can see the heterogeneity within the chromosomal structure itself. It encodes for ribosomal RNAs, denoted here as rRNA, messenger RNAs, denoted here as mRNA, repetitive intervening sequences that may or may not encode for any functional protein or RNA. There are also tRNA genes, shown here, and other large nuclear organizer genes, shown here. Keep in mind there are large stretches along a chromosome that do not encode for any transcribed or any translated protein product. There are also intervening highly repetitive sequences which help in recombination during DNA replication. The superstructure of DNA is shown here. There is a vast amount of double-stranded DNA in the human body, and it is arranged in chromosomes in the following structure. In short, it's supercoiled around proteins known as histones. There are several different types of histone proteins, and within the nucleosome core, which consists of 8 histone proteins, there are 2 of each of the following. There are 2 H2A, 2 H2B, 2 H3, and 2 H4 histone proteins. Within the DNA linker region, shown here, this includes histone H1. These nucleosome cores are then coiled around each other to generate a solenoid structure. This allows for tight packing and condensation of large amounts of double-stranded DNA. Histone proteins can be modified by several different modifications which will either enhance or deter this supercoiling formation. Acetylation is one of the most common modifications to histones. Acetylation of a histone reduces the positively charged lysine residues found on the exterior of histones. DNA, being negatively charged, binds tightly around positively charged histones. If a histone is acetylated and the positive charge on a histone is reduced, this allows for the DNA to become free from a histone. This opening or dissociation of the DNA from the histone allows for greater access for the transcriptional and DNA replication machinery. Conversely, methylation of a histone is often associated with transcriptional repression because of tighter association between the DNA and the histone. We'll look at specific example of these interactions when we look at regulation of transcription and translation. Like protein, DNA has a structure and function that's highly linked. Your book outlines a series of experiments that facilitated the elucidation of both the double helix and the concept of base pairing between adenine and thymine bases and guanine and cytosine bases. In short, there is an equal number of adenine and thymine and guanine and cytosine in all DNA strands. This was the first indication that these formed base pairs with one another. The structure of DNA in this double helix provides protection from the bases. Remember, the bases are what encodes our genetic information, and it is necessary to maintain a pristine copy to ensure genetic stability. The strands are also self-complementary, and replication of DNA is semi-conservative such that there is always a parental strand and a new strand. Consequently, genetic information is highly conserved. If we look specifically at the backbone structure of DNA, shown here, nucleotides are linked through a phosphodiester linkage, where a phosphate group, shown here, links with a methyl group on the sugar. This is your sugar-base linkage. Base, shown here, sugar here, phosphate group. This unit a sugar, phosphate, and a base is considered a nucleotide base. This phosphodiester linkage is integral to the synthesis of DNA. The hydroxyl group here at the 3' end allows for extension of the DNA strand. Your bases, guanine and adenine, comprise the purine bases. Cytosine and thymine comprise the pyrimidine bases. We'll discuss these structures in detail in a few slides. Shown here is a nomenclature for our nucleotide bases again. Shown here is a nucleoside. This consists of a sugar associated with its appropriate base. The sugar group shown here is that for RNA bases. You can tell this because of the hydroxyl group shown here at the 2' carbon. This makes RNA bases extremely susceptible to denaturation and to cleavage. Consequently, this hydroxyl group at C2 is removed to generate deoxybases for DNA synthesis. The addition of one phosphate group, shown here, generates a nucleoside monophosphate, denoted NMP. The addition of two phosphates generates a diphosphate group, or NDP, and likewise, the addition of three phosphates generates a trinucleotide phosphate. Again, a sugar and a base is referred to as a nucleoside, whereas a base, a sugar, and a phosphate group is referred to as a nucleotide. This will become important when we talk about synthesis and salvage. Likewise, the free bases, denoted here, have slightly different nomenclature than a base associated with a sugar, denoted here. Adenine is a free base, whereas adenosine is the base adenine associated with a sugar. Guanine, associated with a sugar, is denoted guanosine, and so forth. Recognition of this nomenclature will be particularly important when we talk about salvage of purines. Now let's look more closely at purine and pyrimidines. As this is the first biochemical pathway we've gone through, let's look and highlight some specific aspects important to the pathways. We will do this for all metabolic pathways that we review. The function of this pathway is to synthesize nucleotides required for DNA synthesis, as well as for RNA transcription. You should be aware of the substrates of this pathway. The substrates required for synthesis of both purines and pyrimidines is listed here. In general, for both pathways, a sugar, a 5-carbon sugar, is required. This charged 5-carbon sugar is denoted PRPP, or phosphoribosyl pyrophosphate. Additionally, the other substrates are required for generation of the ring structure of the base. In the case of purine synthesis, both glycine, glutamine, aspartate, and folate, denoted here as THF, tetrahydrofolate, contribute to the synthesis of the ring. In the case of pyrimidine synthesis, which is a smaller base structure consisting of a single ring, glutamine, carbon dioxide, and aspartate primarily contribute to that ring. These pathways can occur in many different tissues, but the primary location is in the liver, within the cytosol of the cell, and the regulated enzymes for each of the pathways are shown here. In purine synthesis, glutamine phosphoribosyl transferase is the regulated enzyme for this synthesis. You can see here it's activated by the presence of PRPP, which is our 5-carbon sugar, and inhibited by the presence of both AMP and GMP. In pyrimidine synthesis, the regulated enzyme is carbamyl phosphate synthetase 2, and that is inhibited by uridine monophosphate. This is just a summary of the two pathways, and we'll look at these in more detail now. Before we do that, let's look at deoxyribonucleotides versus ribonucleotides. Shown here is a ribonucleotide base. You can tell that because of the hydroxyl group on the C2 ring structure. To convert this ribo base to a deoxyribo base, you must remove this hydroxyl group. This reaction is facilitated by an enzyme, ribonucleotide reductase. Bases are synthesized as the ribonucleotide form. This is because the majority of the genetic components in the cell are present in the RNA form. Ribonucleotide reductase, shown here, uses thioredoxin as a cofactor and NADPH to remove the hydroxyl group. The regulation of this enzyme is twofold, and we'll look at that in the next slide. Also of note, this conversion from the ribo to the deoxyribo base structure happens at the diphosphate level. The enzyme ribonucleotide reductase is regulated at two sites within the enzyme. It has one site which regulates enzyme activity as an on or off, and an additional site that regulates the substrate specificity. There is a single enzyme responsible for the conversion of all of the nucleotide bases from the ribo form to the deoxy form. The enzyme is activated by the presence of ATP. High levels of ATP bound to the site that regulates enzyme activity initiates enzyme activation. If DATP or deoxy-ATP is bound, the enzyme is inhibited. The substrate specificity shifts based on which nucleotide is bound to the effector site. This is summarized in the table here. Let's look at an example. In this case, if ATP or deoxy-ATP is bound to the substrate specificity site, UDP is the preferred substrate for the enzyme. We can tell the enzyme is active in this example because ATP, listed here, is bound in the activity site. If deoxy-TTP is bound to the substrate specificity site, the preferred substrate for the enzyme in that case is GDP. Again, the enzyme is active in this case as ATP is bound to the activator site. You should be comfortable with the transitions between substrates based on which nucleotide is bound to the effector site. Now let's look at the synthesis of PRPP, or phosphoribosyl pyrophosphate. This is the charged 5-carbon sugar required for the synthesis of both purines and pyrimidines. Ribose 5-phosphate is synthesized from a pathway termed the hexose monosphosphate pathway, which we'll discuss in later modules. Keep in mind that ribose 5-phosphate is initially metabolized from glucose. Here, ribose 5-phosphate is accepted by PRPP synthetase, the enzyme that converts it to 5-phosphoribosyl pyrophosphate. The enzyme PRPP synthetase requires magnesium as a cofactor. It's also inhibited by purine ribonucleotides and is activated by free phosphate. Let's pause here and look at a few questions. The first question refers to the acetylation and deacetylation of histones. Try and finish this sentence. Let's first remember what acetylation does to a histone molecule. Acetylation on lysine residues decreases the positive charge on lysines. This decreases the affinity of histones for DNA, allowing the DNA histone complex to become unraveled. Consequently, deacetylation of a histone will increase the positive charge on lysine residues, increasing the binding between histones and DNA. The correct answer here is A, increase. If this is unclear, you should review different modifications to histone proteins. Acetylation and methylation are critical. Now let's look at this question. Again, see if you can fill in the blank. Let's remember ribonucleotide reductase. This is the enzyme responsible of generating the deoxy form of all of the bases required for DNA synthesis. If you recall, it has two effector binding sites. The first, and simplest, requires binding of ATP for activation of the enzyme. So when ATP is bound, the enzyme is active. In this case, the question is asking, under what conditions do we get synthesis of deoxyguanine diphosphate? So the preferred substrate we're looking for is GDP. To facilitate the synthesis of GDP, what has to be bound in the substrate specificity site? If you refer to the table, the answer here is D, deoxythymidine triphosphate bound to the substrate specificity effector site will facilitate the synthesis of deoxy-GDP.